Yeah, good evening. Welcome. Today we are going live. Metabolism, oral health group, biochemistry 2. I've seen one person just on the charts tell me hi. Today's discussion is for the oral health, metabolism. Betty, tell them to keep on logging. Thank you so much. Let's log in. YouTube channel, just go to my channel. I would like to revise on metabolism, the final revision before we can do exams. Just tell me in the chats, hi, oral health. Good, Betty, thank you so much. Tell them to log in. We'll give them like 10 minutes, if more, for everyone to log in if they're having challenges. Thank you so much. Thank you so much. Hi, thank you so much. Thank you so much. Just say hi as we log in. Thank you so much, everyone. Let's log in. It's for metabolism, oral health, please. If you feel it's not the right one, just log out, but we're having revision with the oral health group. Hi, thank you so much, Humeria. Thank you so much, Zach. Thank you, Betty. Let's log in. We are giving it 10 minutes. I think 10 minutes, even more. We'll do 5 minutes because I don't want us to take much time. Since the students can be exhausted, we already do it at night. So we'll have 1 hour maximum. And I hope we'll be able to get something. So if we say by 10 past 9, good. Thank you so much. Just say hi as we log in. Let the students log in. Just tell them we are on YouTube channel. We are live, so they can log in. And the session is for metabolism. I know the students are different to subscribe. It could be biochemistry introduction. It could be genetics. It could be metabolism. Hi, Al-Bashir Abdi. All are welcome, but today, Savita, welcome. I'm not a sir, I'm a madam. Hi, Liwel Masharia. So today we are with the oral health group. We are doing metabolism revision in preparation of the students for the exams. So we have to prepare them for exams, how they need to answer questions, what they need to do. Yes, Happy New Year, Savita. Happy New Year. Oh, Butu. Yes, thank you, my dear. I'm still here. If you miss my face, you just come and log in. You're welcome. Butu. And maybe the nursing students can also log in. Butu, you can tell Karen Okwanyo. Yes, good evening to you too. Hi, Savita. We can talk that one later. For now, oh, Butu, you missed me. I also missed you too, my dear. You can communicate to Karen, please, so that they can also log in because they're doing metabolism. We are doing a final revision today for metabolism. Butu, just communicate to them. You missed me. I also missed you too, you people, but then we are still together. Hi, Imos Mint. Yes, hello. Hi, everyone. Let's just keep on logging in. We'll give it until 10 past 9. Then we'll start. I already sent the questions to Betty, which she shared with the class. Please let them know. Go to just tell Karen. If you tell Karen, I know most of them, Octavian, welcome. Most of them subscribe. If you subscribe, you'll see that I'm live. So just try and talk to them so that they can come. So we are going to discuss metabolism, possible questions. I already shared, so that when you see them, you know what to answer. Yeah. No, I'll not answer those questions of which country I'm from. Kindly we are revising. So it's a revision session. So the other questions can come later, not even this time. Yes, thank you so much, Imosint. That's okay. So let's just keep it for revision, please. Not any other questions, because that one won't work much. So let's just keep it for revision. And then those other questions can go to any platform, but not here. Betty, mobilize them so that they can log in through the channel. We are giving it until, I say 10 past, but maybe some students are having challenges, so we have to wait. So we give them time. Betty, Vutu, as you tell Wakina Karen, kindly just try to talk to them. And I don't know how I'll share with Karen the sample questions, because we're just going to mention question number 1, question number 2, question number 3. But let her tell me if she's on board, then I'll be able to send, because I'm already on my laptop. I can forward to their class email if they're available. Then we can discuss together, because I know it can be helpful as well to my nursing students whom I taught Biochem 1. So let us just be together in this and see how we can be able to answer the metabolism question in any case they are brought in the exam or in our Cat 2, which we are going to do in the oral health. I hope it will be helpful to the students. So let's keep on logging in. Betty, I'm waiting for a response from your end, and Vutu as well. If she found Karen, then they can be able to join. Or any group of, are they May 2024? No, September. Any group of nursing, September 2024, they can also join, the ones who are doing Biochem 2. If they're there, Wakina Maureen, you can tell me that you're there. You just say hi, and the course, I'll be able to know that we are represented. So let's keep on logging in. We are at 19 and it goes to 17. I give it time before we can start, but let us just log in. Today it's a revision for metabolism. For the previous days, it has been for genetics, and it is a week to exams. The exams are almost starting, so we need to prepare well by looking back the whole semester. Whatever we've learned, if such questions come, how can we tackle those questions? So that is the essence of this discussion. So those who are asking those questions, I'm not going to answer, I'm just going to ignore, because the platform is for revision, please. Just keep it respectful, please. That is my plea. Make it respectful. Huh? We're just going to revise, and then maybe later on in another platform you can ask such. So Betty, I'm waiting for feedback. I'm waiting for the feedback. Betty, oral health, are they trying to log in? Thank you so much. Let us keep on logging in so that we can start. I'll still wait because it's already like 13 past, almost quarter. Yes, Frey, hello. Let's keep on logging in. Betty, you will tell me when you are ready. Betty, you are the one who will tell me when you're ready. So just tell me when yes, I can see they're increasing, because we are almost 60, we are 64. Oral health are 64. So a good number, maybe of 40 will start, or even half of it will start. Because as we start, we can start now, correct? So Betty is already telling me, as people keep on logging in, we'll be able to keep on revising as they log in. Huh? So, Betty, I hope you shared with the students. Just let me know. Shamsa, hello. Thank you so much for being there. Please feel at home. So, Betty, I shared the questions with you, and I hope you shared with the students, right? So I'm just going to mention the question number, then we'll be able to, thank you so much, Betty, thank you so much. We'll be able to see on how we need to answer such questions. It's going to help in preparation for exams. I said the reason for this channel is not me, but for us and the students, to help the students understand biochemistry. And that's why we're talking about biochemistry simplified. We are trying to make it simple. However much people think it's difficult, but it depends on the attitude. So I say we remove the negative energy and we put the positive part of it so that it can give us the effort to pass. So in each of those questions, I didn't separate them to sections. Like this is lipid metabolism, this is carbohydrate, this is protein, but then they're random. So I'm just going to mention the question based on what Betty shared. So question number 1 says, mention where beta oxidation takes place and list the steps involved in beta oxidation of fatty acids, and you state the enzymes involved in each step. Remember, beta oxidation talks much about degradation of fatty acids. Whatever you did in biochemistry 1 was you looked at the fatty acid structure being the building blocks of those lipids. So when you talk about beta oxidation, we are going to look at an overview of beta oxidation, how it happens, before we can answer that question. So we said at the point of beta oxidation, you are talking about degradation of those fatty acids. And if you look at the structure of the fatty acid in biochemistry 1, to the left-hand side, we had the methyl end. To the right-hand side, we had the carboxyl terminal end. And whatever you also saw was, we had to classify them as saturated and unsaturated. And for the saturated, whatever you discussed was, there is no double bond. For the unsaturated, there were double bond. So when you talk about the beta oxidation, we are going to discuss much on the saturated fatty acids. But we start with the even chain fatty acid because there is also odd chain fatty acids. So for the even chain fatty acids, we said degradation of those fatty acids, the even chained, will give us acetyl-CoA. But if you look at the odd chain fatty acids, you are going to get propionyl-CoA and an acetyl-CoA because it will reach a point now where you have 5. You know, you've been breaking it down 2-2. Those 2-2 are the acetyl-CoAs. So when you reach at the point now where there are 5, it means it will give you propionyl-CoA and acetyl-CoA. That is for the odd chain fatty acids. And the propionyl-CoA will find its entry into the TCA cycle because the final product which will be formed will be succinyl-CoA. So let us focus on the even chain fatty acids first. And I mentioned that before the beta oxidation takes place, remember, I told you to go and read it, but I already explained whatever beta oxidation is all about. So before beta oxidation takes place, there are some of the processes which has to take account. Yes, Ochieng Sandy, the live session, that is the good thing with YouTube account. It is going to save itself and you're going to see it. So don't worry about that. What I'll do is I only have to put it like public. If I put private, the students won't see. And that will be unfair because the students are the ones who have to reach for it. So I'll just put in my settings public, and then it's like it will be visible for all the students who have subscribed. You'll be able to see it. So don't worry, Ching. It will show. And immediately after 1 hour, you'll be able to see it because it keeps on, it will process after I'm done but that it will show. Thank you so much. So let us just concentrate, and then afterwards you'll be able to see the video for discussion or for revision. So whatever I was saying was, before these fatty acids get broken down, before the degradation process, beta oxidation is the degradation of the fatty acids. So we are saying the first step is we have to activate our fatty acid. Remember I said the structure of the fatty acid has the carboxyl terminal end to the right hand side. There is a COOH. But where does the name acetyl-CoA come from? So it means it has to be activated by addition of a CoSH group, where the thiokinases do the work, because there has to be a form of ATP. So it has to be activated first. The first step before beta oxidation takes place, you have to activate the fatty acid. That is addition of CoSH group. After activation, you know, the activation takes place in the cytosol. After activation, then there has to be a carrier protein called carnitine. The name carrier, it has to shuttle our activated fatty acid from the cytosol to the inner mitochondrial membrane, or to the mitochondria, to be precise. Because those are two different organelles. The cytosol is where activation takes place, but then it has to be ferried to the mitochondria for beta oxidation to take place. So the carnitine as a protein will have to ferry our activated fatty acid. After it has been ferried into the mitochondria, then definitely now degradation or beta oxidation takes place. And at the point where beta oxidation takes place, it only involves 4 steps. The first step, generation of NADH. Already dehydrogenase is involved. The second step is hydration. The third step, generation of FADH2s. Remember the NADH and FADH2s for the assignment which I gave, just discussed the ETC, the electron transport chain and oxidative phosphorylation process. They are used as electron carriers. The NADH from the TCA cycle, the FADH2 from the TCA cycle, the NADH from the beta oxidation, the FADH2 from beta oxidation. They are used as electron carriers in electron transport chain, which also generates ATP. See how we talk about metabolomics, how the lipid metabolism connects with carbohydrate metabolism with protein metabolism. They need one another. That is how important biochemistry is, the chemistry of life. We start with the biomolecules, introduction to the biomolecules, including structures. Then we dive into metabolic pathways, including the enzymes, because if they are defective, then it leads to a disorder or a disease. So whatever we are saying, before degradation of those fatty acids, I have to be clear in this, there is activation process. After activation, activation is addition of CoSH group. After activation, we need to shuttle it, or to transfer it, or to transport it to the mitochondria. And the carnitine will do the work. After transporting it to the mitochondria, the 4 steps will take place. Beta oxidation involves the 4 steps. They talk about oxidation. It involves a dehydrogenase, the first step. But NADH is generated. First step, don't even try to interchange them. That's how it goes. NADH, it's a dehydrogenase. Second step, hydration. Third step, another oxidation. Second oxidation, another dehydrogenase. They fall under the class of oxidoreductases. That is the class of those enzymes, which you'll always do in one. But then, at that point in step 3, FADH2 is generated. And then the last step is thiolysis, breakdown. If we started off with C14, C14 was myristate. C14 was myristate. ZC tribal, can you just log out? Can you log out kindly? Because we are discussing serious things here. You are not discussing me. We are discussing metabolism. Kindly log out. Now, what I'm saying here is, at the point where we have the fourth step, we are talking about thiolysis. We are breaking down, if you started with C14, which is myristate, or laurate, or lauric acid, C12, at the point of step 4, you are 2 carbonless. Because if you look at the structure of acetyl-CoA, it is 2 carbons. So at that point, you will be having C12 and then acetyl-CoA. Then it goes again to step number 1 until you will have 7 of the acetyl-CoAs if you started with 14. 6 of the acetyl-CoA if you started with 12. 8 of the acetyl-CoAs if you started with palmitate. So it involves those 4 steps: dehydrogenation, hydration, dehydrogenation, and lastly thiolysis. So we talk about the type of metabolic pathway for beta oxidation is a spiral type of metabolic pathway. You can see the same enzymes. It's like in a spiral manner or a spiral pattern until at last you have the acetyl-CoAs. So breakdown of the fatty acids, is going to give us the acetyl-CoA. And I mentioned the beta oxidation or breakdown will take place in the mitochondria, but the activation takes place in the cytosol. The carnitine has to transport it into the mitochondria for them to get prepared for beta oxidation. So for those 4 steps, that is what involves the beta oxidation. And in each of every step, a reaction takes place. After the 4 steps, we are reducing our fatty acid to carbonless. So for the even chain fatty acids, we are going to get acetyl-CoA. For the odd chain fatty acids, you start by cutting 2, 2, 2, 2 until at last you have 5, because 5, you know, it's now not even. If it's even, it will end up with acetyl-CoA. So you have 5, 3, 2. 3 is the propenyl-CoA. 2 is the acetyl-CoA. But then the propenyl-CoA is broken down into methyl malonyl L, methyl malonyl D, which at last forms the succinyl-CoA. Succinyl-CoA is an intermediate of the TCA cycle. So it will go to the TCA cycle to generate the reducing equivalences. And you know at what point the succinyl-CoA will work. It will just get the NAD-dependent enzyme, which is malate dehydrogenase, because the alpha KGs and the associated dehydrogenase are before the succinyl-CoA. So for the odd chain fatty acid, we are going to get propenyl-CoA together with acetyl-CoA. For the even chain fatty acid, we are going to get acetyl-CoA. The acetyl-CoA, which is generated from the degradation of those fatty acids, can go to fatty acid synthesis, can go to formation of the ketone bodies, or it can also go to the TCS cycle to generate reducing equivalences. So I've tried to give an overview of what a beta oxidation is all about. So mention where the beta oxidation takes place. You can just say the mitochondria, you will still get your mark. List the steps involved in beta oxidation and the enzymes. So the first step has to be the NADH generation step. A dehydrogenase is involved there. I told you, if you can't remember clearly the name, but then the class of enzyme is correct, just give me the class of enzyme. Whether it's a dehydrogenase, it's a kinase, I'll give you a mark. Because sometimes you can master the structure, but getting the correct name, you talk about beta acyl CoA dehydrogenase. That one, maybe the name will be difficult, but being a dehydrogenase, I'll give you a mark. And I've always given the mark, even for the cat which you saw. Because even for the student to think of the correct class of enzymes is already, he or she has done a lot of work. So if the class of enzyme is correct, I'll give you. So the first one is a dehydrogenase, but it has to involve the NAD plus dependent enzyme, because at that step number 1, NADH is generated. Please don't confuse it with FADH2. FADH2 will come at step number 3. So step number 2, we have hydration, and step number 2, we have hydration. Step number 3, we have oxidation, where now we have a dehydrogenase. And lastly, we have thiolysis, where the thiolase as an enzyme will break down our fatty acid into acetyl-CoA and 2 carbon less. So that is the basics. So just mention the enzyme, mention where it takes place, and mention the step. That's fine. Using structures, we are at number 2. Using structures, if you have questions in the chat, just let me know. I'll be able to answer. Using structures, show the steps involved in fructose entry into glycolysis. We are following the questions which we shared. Hi, so I can see people are still logging in. Just log in as we proceed. I might not answer some of the highs because we already started, but it's okay. Let's stay on board. So show steps involved in fructose entry into glycolysis. So that one tells us much, that maybe you took food which had sucrose at that point, glucose, and we said it has to start with digestion, then proceed with absorption. After absorption, oxidation of those sugars takes place. So we talk much about glucose oxidation in glycolysis, but now sucrose, the sugar which we use in the morning, has a component of alpha D glucose and beta D fructose. So we are asking ourselves, where do the fructose go? Because it also has to provide energy in form of ATP. So we need to find an intermediate of fructose as well, which has to be seen in the glycolytic pathway. So we have to convert it to one of the intermediates of the glycolytic pathway. That is, after in the small intestine, the sucrase will break it down or will digest it into those 2 simple sugars. So at that point, glucose will go straight to glycolysis. But fructose, what we saw could be 2 options. Fructose has to be converted to fructose 1-phosphate by the fructokinase, specificity of enzymes to be precise. Fructokinase, remember the kinase, as I said, they need magnesium ion or manganese in their active site. But then it has to be an ATP coming in, out as a diphosphate, because 1-phosphate will phosphorylate our fructose at carbon atom number 1. It will form fructose 1-phosphate. Then the aldolase is still aldolase. It will break it down. And then the upper part will have the dihydroxyacetone phosphate. The lower part will be glyceraldehyde. But glyceraldehyde is not an intermediate of the glycolytic pathway. So the triose kinase will phosphorylate glyceraldehyde for us to get glyceraldehyde 3-phosphate. Then we say the arrow goes to glycolysis. That is what we learned in class. But we said another alternative is at the point where it forms fructose 1-phosphate. I told you someday I'll use my whiteboard, which is there at the back. You can see my whiteboard is there. We improve step by step. I'll be able to explain this on the board because it's easier if the students can see on the board. So for today, you can just have a pen, maybe you write down. The recording will still be available as well so that you can help it for revision. So I'm saying the second option was at the point you are forming fructose 1-phosphate, mutase will do a shift. That is the second option. Mutase will do a shift. We will shift our phosphate from the first carbon of fructose to the sixth carbon to form fructose 6-phosphate, which is an intermediate of the glycolytic pathway. Simple as that. That is how the entry of fructose to glycolytic pathway works, or how fructose provide energy in form of ATP, how it enters the glycolytic pathway. Question number 3 says, using structures, show how asparagine connects to TCA cycle. Using structures. Very simple. What they're asking there, do you understand the glucogenic amino acids? We define them as amino acids whom their degradation products will form pyruvate or any of the intermediates of the TCA cycle. And we saw alanine, serine, glycines, which was again the simple amino acids will give us pyruvate. Especially alanine is just one step. And then you have pyruvate. So we name them as glucogenic. And even the ones which were giving us the intermediate of the TCA cycle were glucogenic. So the best example we had was aspartate being converted to oxaloacetate. And then glutamate was converted to alpha KG, which are intermediates of the TCA cycle. So that makes them to be glucogenic amino acids. But what of asparagine and glutamine? Simple. Aspartate was forming from asparagine, the enzyme asparaginase, removing the amine group. So you start with asparagine. From asparagine, you know the structure of asparagine is just aspartate, but you add the NH2 at the R side chain. So the asparaginase is going to remove the amine group on the R side chain. You're going to have aspartate. Then formation of aspartate to oxaloacetate. It means the oxaloacetate will enter at that point where the acetyl-CoA is available from the conversion of pyruvate via the pyruvate dehydrogenase complex of enzymes. Then it's going to form citrate. So that's how it connects. Asparagine has to be converted to aspartate, then aspartate to oxaloacetate. So you just mention at that point where it forms oxaloacetate, it is going to condense with acetyl-CoA to form citrate. Then you stop. That's how it enters the TCA cycle. But I say using structures. So it finds when in biochemistry 1, you've learned the structure of amino acids. You've seen the structure of oxaloacetate in gluconeogenesis. You've already known the structure of oxaloacetate in TCA cycle. So it won't be difficult. So you just mention, after formation of oxaloacetate, you mention it enters the TCA cycle where it combines with acetyl-CoA to form citrate. That's how simple it is. State the roles. Number 4, state the roles of the enzymes transaldolase, pyruvate carboxykinase, the alpha KG dehydrogenase phenylalanine hydroxylase. I said if I bring such a question, just tell me it converts this to this. You know, like the transaldolysis, they are involved in the transfer of the 3 carbon molecules from a keto donor to an aldose acceptor. That's already enough. Some people are also writing the equation where 7-pseudoheptulose is involved together with the glyceraldehyde 3-phosphate. So if you transfer 3 carbons from the pseudoheptulose 7-phosphate, you are going to form 4 erythrose. Is it erythrose? Yes. Erythrose and fructose 6-phosphate. That is still okay, if you can remember it well. Because for me, I'll see there are 3 carbons which are cut at the upper part. For a ketose donor, you know, acetoheptulose is a keto. Heptose, 7 carbons having a keto group. You will cut the upper 3, mount it on top of glyceraldehyde. It will form fructose 6-phosphate. The lesser three carbons will remain to form erythrose. So that is still okay. But the simplest way is tell me how it transfers the 3 carbon molecules from a keto donor to an aldose acceptor. Then the transketolase was transferring 2 carbon from a ketose donor to an aldose acceptor. So just tell me what it converts from this to this. Don't go to a lot of stories. It's going to take your time for no reason. And don't mention for me the pathway and you leave it like that. You know, even if you mention the pathway and I didn't say you mention it, I'm not going to give you any mark. Yes. So the second one is pyruvate carboxykinase. So the first one, if you can remember the pentose phosphate pathway, that's where we get the role of that enzyme in carbohydrate metabolism, of course. Pyruvate carboxykinase, you've seen it, the first bypass of the gluconeogenesis. So in the first bypass of gluconeogenesis, we have the formation of oxaloacetate from pyruvate, that is carboxylation. Pyruvate carboxyl doing the work. But then that first bypass has 2 steps. We have the formation of PEP from oxaloacetate, where this enzyme is involved. The pyruvate, phosphoenol pyruvate carboxykinase. That is the full name. It's not pyruvate carboxykinase. Phosphoenol pyruvate carboxykinase. I think I left something. So it's the one which catalyzes the conversion of oxaloacetate to PEP. That's already enough. Alpha ketoglutarate dehydrogenase. You can remember the TCA cycle where the alpha Kg is involved in the generation of NADH. Just say whatever it converts from this to this. It's already enough. Don't talk about a lot of things. The phenylalanine hydroxylase, you know it as well. So where the phenylalanine is converted to tyrosine, because tyrosine has the hydroxyl group down there. There's anhydration there. So it is the phenylalanine hydroxylase. Conversion of phenylalanine to tyrosine. Point number 5. We are at question number 5. Please let us refer to the questions to make work easier, because I promise to take shorter time. Differentiate between ketogenic and glucogenic amino acids and give one example each. Look at how the marks are swallowed. You get them in bulk. What I mean here is define ketogenic amino acids. The ones whom their degradation products will give you ketone bodies. And if you talk about the ketone bodies, we have the beta hydroxybutyrate, we have acetoacetate and acetone. Then glucogenic, their degradation products will give us pyruvate and any intermediate of the TCA cycle. Just by giving one example, you will just get 1 mark for the example, 1 mark for definition, 1 mark for the example, 1 mark for the definition, 4 marks. So you know them. Question number 6. Using structures show only, I said only, show only the ATP utilization steps in glycolysis. I have to specify the pathway so that you don't confuse the students. ATP utilization steps using structures. We are going to use structures. So we are saying that the ATP utilization step, put the arrow, go inside as ATP, goes out as ADP, then you will know. We are talking about stage 1 of glycolysis. Step number 1 and step number 3. Using structures. Show only ATP utilization step. The step where hexokinase is involved and the step where we have the phosphofructokinase. So the conversion of glucose to glucose 6-phosphate, that is what I need. Don't even write the phosphoglucoisomerase one. No. I only need the structure for glucose and glucose 6-phosphate. Hexokinase involved there. ATP going in and out ADP. For some, there were no direction. If you don't have direction, I just round it. Because if there is no direction of the arrow, where the arrow goes to, it becomes difficult to know whether you are talking about the ATP utilization or ATP generation steps. So just make the arrow clear. It has to be clear that it comes in as ATP, goes out as a diphosphate. And then I said step number 1 and step number 3. In step number 3, the formation of fructose 6-phosphate from the glucose 6-phosphate makes the phosphofructokinase to catalyze that step by adding a phosphate. So it goes in as ATP, out as ADP, and it is going to form the fructose 1,6-bisphosphate. Those are the steps I need. 4 marks. Number 7 says, list the enzymes bypassed in gluconeogenesis and match them with their counterparts in glycolysis. List. If you look at the bypassed steps, we talk about the bypass because the enzymes in gluconeogenesis change. The irreversible steps, they change. The reversible ones remain the way they are. By definition, gluconeogenesis, you are synthesizing glucose from the non-carbohydrate precursors. And if you talk about the non-carbohydrate precursors, you're talking about amino acids, you're talking about glycerol, you're talking about lactate, all those non-carbohydrate precursors. That is gluconeogenesis. So we're talking about the bypassed reactions. So instead of the arrow going towards formation of pyruvate as it is in glycolysis, the arrow goes towards the formation of glucose. So they want you to match. In glycolysis, we have hexokinase, but now that is the first step of glycolysis. It is the last step of gluconeogenesis. So you'll talk about 2 columns, glycolysis, gluconeogenesis. You will put hexokinase there. Then in gluconeogenesis, you'll talk about the pyruvate carboxylase and the phosphoenol pyruvate carboxykinase. Then you go to another one. You say the bypass, second bypass in gluconeogenesis, where we have fructose 1,6-bisphosphatase. In glycolysis, it is the phosphofructokinase. Still the last bypass, bypass number 3, glucose 6-phosphatase. It is the first step in glycolysis. You'll have hexokinase. That's how simple it is. And what did they say? 3.5, 1.5, I think it was definition of what gluconeogenesis is, which I can't bring nowadays. What is gluconeogenesis? I will not bring that. You will just add it up. When I talk about how the amino acids can connect with gluconeogenesis, that is the connection of the amino acid metabolism with the carbohydrate metabolism. So at that point, that's how I can set it. But I will not set definition of any of the pathways. I think that is the 1.5 which was missing because it has to be adding to 5 or 4 marks. Using structures, show the steps involved in stage 2 of glycolysis. stage 2. You know, the stage 1, we were destabilizing our glucose, such that at the end of stage 1, we had fructose 1,6-bisphosphate. Stage number 2, we were generating the trioses, because remember, whatever happens is we are going to generate 2 pyruvates by starting with a D-glucose. So the generation of trioses happens in stage 2. And then stage 3, it brings about the final objective of the glycolytic pathway to provide energy in form of ATP. So stage 3, it's for ATP production. So at this point, we're talking about stage 2. We are talking about the interconversion of the trioses. We are talking about the cleavage, the aldol cleavage which takes place at step number 4. Step number 5, the interconversion of the ketotriose and aldotriose to a ketotriose. So whatever we are saying here is stage 2 has the aldolase breaking down of our fructose 1,6-bisphosphate to dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. Then the interconversion of the two using the triose phosphate isomerase. That is stage 2. Simple. Give me the structures. You'll get all the marks. We are saying which one again? We are at number 9. Using structures, show the one-step degradation of alanine. The one-step degradation of alanine. Alanine, you know the structure. I said we have to answer it correctly, the way we understand it. Alanine is very simple structure. It finds when you know that alanine falls under the aliphatic amino acids. Just as simple. The R side chain just have CH3, a methyl group. Simple as that. You already know the structure of pyruvate in glycolysis, because I told you in metabolism, if you don't understand how the glycolysis takes place, the glycolytic pathway, then the other pathways will be difficult. So it finds when you already know the structure of pyruvate. Imagine how I'm trying to make it simple. So the one-step conversion of alanine, you'll get a full mark for the structure, full mark for the pyruvate and alanine. And then the group which carry the amine group, you can see the glutamate and alpha Kg here. They carry the amine group. There is amino terminal end there. When that amine group is removed, we talked about the urea cycle now. That is where it goes. Because if it's left there, it's going to be toxic. So we are talking about the glutamate and the alpha Kg being the carriers of the amine group, which is found when those amino acids are broken down. So we are converting alanine to pyruvate, the alanine aminotransferase, a very important enzyme to detect liver damage. The ones who are doing clinical biochemistry. Important enzymes to know whether the liver is damaged. Aspartate transaminase, aminotransferase. And then we also have alanine, very important. So we have the alanine, aminotransferase, and then we have the one-step conversion of alanine to pyruvate. That's what I need. Because I know you're always familiar with the alanine structure, you're familiar with the pyruvate, and you can be familiar with the enzyme. So that's how easy it is. Just give me whatever I've asked and you'll get the mark. Whatever you're not asked, don't answer. Just answer whatever you are asked. Where does beta oxidation takes place? In the mitochondria, of course. State the 4 steps involved in beta oxidation. You can see how my setting is If you see the pattern, you'll know. Beta oxidation steps, just been told the steps because that's already 5 marks. 1 mark, you just mentioned the mitochondria. The steps. Step number 1, you talk about oxidation. They didn't even ask the enzymes. Step number 2, hydration. Step number 3, oxidation. Step number 4, thiolysis. Simple. Write short notes on phenylketonuria. Most of the questions I ask, I say using structures, describe a pathway, using structures, describe a pathway. But sometimes when it comes to the diseases which you need to discuss as a result of defective enzyme in any of those pathways, then you just need to explain this enzyme converts this to this. Now this is not converted to this, then it leads to this being high, and then that is how the condition is found. So for the phenylketonuria, from the name you can see, the ketones are detected in the urine and in the blood plasma. So what happens here is you start by saying phenylalanine is not converted to tyrosine because the enzyme phenylalanine hydroxylase is defective. So the phenylalanine gets transaminated to phenylpyruvate such that there'll be high level of phenylpyruvate, phenylactate in the urine and plasma. Then you can talk about the characteristics. It can lead to slurred speech. It can also lead to mental retardation, which might lead to death. So if such kids are born at the time they are born with such conditions, it might lead to their death. So before you talk about the characteristics, first explain what brings about that. Which enzyme converts what to what? At a certain point where we can see the disease, then what is in high levels? You know, in clinical biochemistry, we talk about the normal level of that chemical component which you're testing in a sample. But if the level is above normal, then there is a problem. That is what brings about the disease. So at that point, we are talking about a defective enzyme. This enzyme should go a normal way of converting our substrate to a product. But now, what is happening here, if the enzyme is defective, there is high level of such a component in any of, it could be the plasma, the urine, the mucus, whichever sample, but it has to be the correct one. So that is how you explain such. So I'm saying which one? We are at number, I think some I repeated, but then it's still okay. We'll still learn. It talks about entry of fructose. You know, I also have to scroll it on my laptop. And sometimes it misbehaves, it hangs. Yeah, so it says, using structures, show how fructose enters glycolysis. So we are not going to repeat that because we already discussed. Yes? So we already discussed that one up there. So you'd be able to do it the same we did it on the upper question. Write short notes on Alcaptonuria. You know, Alcaptonuria is as a result of homogentic acid. That is where we talked much about if the kid's diaper is exposed to air, after 30 minutes after, it will turn dark. For adults, there are some deposits. In the intervertebral discs, there's some deposits. If you do the test, you'll find the black deposits in there. So the change of the urine color to black when exposed to air, you know the normal color of urine, and now when the urine there is a problem, then definitely when it's exposed to air, then it leads to the urine turning dark. But then this is as a result of homogentic acid, which is defective. So at that point, After you've explained the enzyme which is defective and the condition which lead to such, then you can talk about the symptoms or the characteristics, and then it can give us mark, but it's only 5 marks. So the most important thing is to mention the enzyme, what could be the cause, and definitely now you come on with explaining much on the characteristics. Using structure still. Now define gluconeogenesis, which I can't bring such. Name key enzymes of gluconeogenesis. The key enzymes are the pyruvate carboxylase, the phosphoenol pyruvate carboxykinase, and lastly we have the glucose 6-phosphatase and fructose 1,6-bisphosphatase. Write short notes on how ketosis results. Write short notes on how ketosis results. We are talking about the ketone bodies. So, if you talk about ketosis in simpler terms, is breakdown of those fatty acids we leave as acetyl-CoA. The acetyl-CoA, which we get from the beta oxidation of the fatty acid, will go to the TCA cycle for the synthesis of the reducing equivalences, the NADH and the FADH2s. If it goes to the TCA cycle and finds oxaloacetate absent, I repeat, the acetyl-CoA, which is generated from the degradation of the even-chained fatty acids, for example, has to go to the TCA cycle to generate the NADH and the FADH2s. If it finds oxaloacetate absent, it will resort to formation of ketone bodies. Or if the oxaloacetate is insufficient for it to combine with acetyl-CoA to form citrate, then it will go to formation of ketone bodies. So the determinant here is oxaloacetate. If the oxaloacetate is not present, the TCA cycle, it will resort to formation of ketone bodies. That is what will lead to the high levels of ketone bodies. Question number? I'm just checking. Fructose. Did I miss something? Oh, yes, I can see. Oh, yeah. Define gluconeogenesis again. State the role of the following enzymes. The beta-hydroxyl acyl-CoA dehydrogenase. Beta-hydroxy-acyl-CoA dehydrogenase. Fumarase. So you know where the beta-hydroxy-acyl-CoA dehydrogenase is? In the lipid metabolism. What I said, as I said in the other ones, just say it converts this to this, full stop. Fumarase, if you look at the carbohydrate metabolism in the TCA cycle. UDP galactose epimerase, if you look at galactose entry. Enol CoA hydrase in the beta oxidation, where the hydration takes place. Just say it converts this to this, full stop, don't talk much. Using structures, outline the TCA cycle. It has the 8 steps. Just outline using structures. Some questions are direct. It will always go for essays. Using structures, do it well from oxaloacetate, acetyl-CoA, citrate, isocitrate. And then we have the alpha Kg, how it goes until you come back to malate oxaloacetate again. So just describe using the structures, the correct enzymes, because it will earn marks. And I told you, the change is only on 1 carbon. You can't change 2 carbons for a structure. For a substrate to get a product, change in 1 carbon. And of course, it could be a removal of 1 carbon because of the decarboxylation which takes place. In each of those steps, I think there are 2 steps where the decarboxylation takes place. But all in all, just put the correct structures, the correct enzymes. If you can't remember the full name, give me a dehydrogenase. I'll give you a mark. If I know it's the relevant one for that step of oxidation reduction reactions. So for the TCA cycle, describe. And then they say its biological importance. We talked much about it generates reducing equivalences, which can also be converted to an ATP. There is an equivalence of NADH to an ATP. There's equivalence of FADH2 to an ATP. But at the same time, the NADH and FADH2s are used as electron carriers in the electron transport chain. Without structures, here I said without structures. Discuss oxidative and non-oxidative phase of the pentose phosphate pathway without structures, which mostly I don't set. I always like using structures. So this was the lucky day for these students. Without structures, describe the oxidative and name the products of the pentose phosphate pathway. So I said in the oxidative phase is where we generate the NADPH. Only in the oxidative phase, based on the need of the cell. If the cell is in need of NADPH for fatty acid synthesis, for the synthesis of the steroid hormones, for lipid metabolism, for integrity of RBCs, it needs NADPH. We will go to the oxidative state phase and stop. Remember, the NADPH is generated in step 1 of the oxidative phase in the pentose phosphate pathway and step 3. In the non-oxidative phase, we are going to generate the riboses, the pentoses, which are going to form the part of the nucleic and the DNA, the DNA and the RNA, the nucleic acids, very important pathway. And then the last bit, which I said, based on who the lecturer is, because for me, I see ATP as the third one, because we are going to find the glyceraldehyde 3-phosphate in the non-oxidative phase, fructose 6-phosphate in the non-oxidative phase, and the pentose phosphate pathway will act as a shunt. So as much as glycolysis is going to bring ATP, it can also act as an alternative pathway, the pentose phosphate pathway. So the products of the pentose phosphate pathway, we have the ribose, the NADPH, and ATP. You know, whatever happens, we start with glucose 6-phosphate, which can come from glycolysis or breakdown of glycogen will give us glucose 6-phosphate. You move to form the 6-phospho gluconolactone. And then the lactonase will be involving the second step. The first step involves the glucose 6-phosphate dehydrogenase. It's the one which controls the whole pathway. And then step number 3, you'll have gluconolactone dehydrogenase, which will form 6-phosphogonate. Then at last, you're going to form ribulose 5-phosphate. The first pentose which is formed in the pentose phosphate pathway is ribulose. Ribulose is a keto pentose. So we have to go a step further in the non-oxidative phase and get ribose 5-phosphate. But then the phosphatase will have to remove the phosphate so that we can have the ribose, which is needed by the nucleic acids. And then the transaldolysis, the transketolysis will be forming there in the interconversion of the sugars. Sorry. The epimerase and isomerase are also involved there. So without structures, describe. Outline the urea cycle and any 2 diseases associated with the pathway. Urea cycle, only 5 steps. Remember, the formation of carbamoyl phosphate. You need the carbon dioxide or the carbonic acid. And then you need the ammonia, NH3, from the digestion of proteins and the degradation of the amino acids. You are going to form the carbamoyl phosphate. And for the carbamoyl phosphate, now you know how the cycle goes. It has to combine with ornithine. You are going to get citrulline, and then from citrulline, you are going to get agenosuccinate. Agenosuccinate, there has to be condensation of citrulline with aspartate to form agenosuccinate. And that is where the citriuria come in. Because there will be a lot of citrulline in the urine. Because there is no condensation. You know, aspartate has to come in and combine with citrulline to form agenosuccinate. Because at the point agenosuccinate is forming via the enzyme agenosuccinate synthase, the lyase does a lot of work. It is going to break down our agenosuccinate into fumarate, which is going to the TCA cycle, and arginine. By the time arginase converts arginine to the next product, urea is released. That is the point where the urea cycle is very important to make the ammonia less toxic for our system. You know, if it's much higher than normal, it's even lead to death. You could see whatever we learned, mental retardation, you can have problem in speaking, but then it can lead to death. So we have to remove, or we have to reduce the toxicity of that ammonia through the urea cycle. So they're talking about the pathway and any of the diseases which are involved. There is a disease which involves the lyase. There is a disease which involves the synthase. I think we looked at example of the diseases, there are like 5. So you can choose any 2. But you have to know which of these enzymes it involves at this step, which when it's defective, then it leads to such. You get. So outline the urea cycle and the diseases. Discuss reaction sequence of urea cycle and add note on disorders. It looks more like the one which we've just discussed. Using structures, discuss fatty acid synthesis. Fatty acid synthesis, if you look at the processes which we saw in degradation, if we had oxidation the other side, here now we have reduction. Where we had hydration, in synthesis we had dehydration. So we just have to get the points right. But remember, in synthesis, we need NADPH, which comes from the pentose phosphate pathway we just talked about. But now we have to condense. We start by condensing, where you are forming the malonyl-CoA, the butyryl. So you keep on condensing until now you have the length of the length of the fatty acid. So you are building until you have, if you are building, you are synthesizing palmitate, we are talking about condensing. So we add to the malonyl, you add another acetyl-CoA, but now we are involving acyl carrier protein. Remember, in degradation, we were involving carnitine. And even in this case, since degradation took place in the mitochondria, synthesis takes place in the cytosol. So they have to be shuttled again using the mallet shuttle. We have to transfer them, citrate, into the cytosol. That's how simple it is. So fatty acid synthesis, I'm at page 2, but some of these are repeating themselves, so I'll be able to move fast now. When you see me, I'm a bit stuck. I'm trying to scroll down. Using structures, outline only the bypassed steps in gluconeogenesis. Only. I said only. And somebody gives you the whole pathway of gluconeogenesis. No. Only the bypassed. Why do you have to make a lot of energy by making the whole pathway of gluconeogenesis? And you've been asked only the bypassed steps. And even to make it simple, you need to tell me bypass 1. I told you, the way we answer questions, it should show that we know what we are doing. Even if somebody comes and asks you or wakes you up in the middle of the sleep and you ask you, oh, in gluconeogenesis, so what do you mean by fast bypass? You just explain it clearly. Bypass 1, put pyruvate, oxaloacetate, put the enzyme there, the structures as well. Remember, the pyruvate carboxylase is ATP dependent. You have the ATP and ADP plus PI. And biotin as a vitamin also there. And then you still have bypass 1. From oxaloacetate to PP, GTP dependent to GDP. Remember, those can also be equivalent to an ATP. Then, at that point, you have the phosphoenol pyruvate carboxykinase. That is bypass 1. Put it clear. You'll get all your marks. Then go to bypass 2. We said only the irreversible steps in glycolysis, the enzymes will change. That is why we are talking about the bypass. Bypass 2, fructose 1,6 bisphosphate converted to fructose 6-phosphate. It means the enzyme is fructose 1,6 bisphosphatase. We are removing a phosphate. Remember, the arrow should go towards formation of glucose. Let us not make mistake, because if the arrow goes towards fructose 1,6 phosphate, I'll give you a wrong, and I'll be very sorry. Let the arrow go towards fructose 6-phosphate from fructose 1,6 bisphosphate. That is bypass 2. Bypass 3, the final part, the final step, where we are synthesizing our glucose. Because remember, the definition goes for gluconeogenesis, we are synthesizing glucose from the non-carbohydrate precursors. So the last bypass is bypass 3. Draw the structure of glucose 6-phosphate, arrow going to glucose. And the enzyme is glucose 6-phosphatase. And remember, in those steps where we are removing a phosphate, a hydrolase is involved. So you have to put water and you release PI. Most students were putting still ATP. So the confusion between glycolysis and the other one should not be there. Some were even putting NADH. I just round it and I put the correct thing. If the enzyme is correct and you put for me ATP, I just cancel the ATP. But in the main exam, I'm not going to cancel anything. I'm just going to cancel everything, because that is the property of enzymes, specificity. You can't put the wrong ones, the wrong cofactors, the wrong coenzymes, and you think you can get an answer correct. No. Let us put the correct things. Let the arrow go in the correct directions. So that is why we are revising, so that we don't make the same mistakes which we did make maybe during the cut, because we still have other chances. At the time of exam, now you are giving it all. So we need to put things right the way they should. So it only talked about the bypassed steps. Steps catalyzed by coenzymes in pathway involving entry of galactose to glycolysis. The coenzymes in galactose entry. You have to give the steps where the galactokinase is involved. You have to give the step where the polymerase is involved. And lastly, you have to give the step where the uridyl transferase is involved. Those are the key enzymes. uridyl transferase, epimerase, and galactokinase. Using structures, outline degradation of any 3 glucogenic amino acids, and it's only 10 marks. So I said, for the glucogenic amino acids, you can give degradation of aspartate and asparagine. Those are already true. You can give degradation of glutamine and glutamate, also already true. You can give alanine. It's a question which can earn you a lot of marks, because you know the one for alanine, you know the one for asparagine, you know the one for aspartate, you know the one for alpha kg, you know the one for not alpha kg, but glutamine and glutamate. Discuss any 2 diseases associated with degradation of aromatic amino acids. So this one do not involve any structure. You are just writing points. You can write albinism, where the tyrosinase as an enzyme is defective. You can write about the PKU, and you can write about Alcaptonuria. In fact, those 3 involve the aromatic amino acid. It's the same pathway. So just write about them. The enzyme, what it converts this to this. The enzyme which is defective, now what does it lead to? What is in high levels? And then what are the outcomes, maybe the characteristics. Explain how ketosis results. I talked about ketosis, the acetyl-CoA, from the degradation of fatty acids. If it goes to the TCA cycle, it finds oxaloacetate insufficient, it will lead to formation of ketone bodies. Outline the pathway for ketone bodies formation. When you see the question, you think it's complicated, but it's very simple. You just start with 2 of the acetyl-CoAs, because I said the ketone bodies will form because the acetyl-CoA, from the degradation of the fatty acid, found that oxaloacetate was not present in the TCA cycle. They will go straight to formation of ketone bodies. So we start with 2 acetyl-CoAs. You're going to form acetoacetate. Then you'll go down the line, acetoacetyl-CoA, then you're going to form the acetoacetate. The acetoacetate will be the first ketone bodies to be forming, and then it will lead to formation of beta-hydroxybutyrate and acetone. So it's also a very simple step. But the acetyl-CoA will come from the degradation of the fatty acids. State 3 examples of ketone bodies just by mentioning acetone, acetoacetate, and beta-hydroxybutyrate. Simple. Some people will always confuse acetyl-CoA as ketone body. It's not a ketone body. Acetoacetate, or acetoacetyl-CoA, acetoacetyl-CoA, or acetoacetate. I know of acetoacetate, acetone, and beta-hydroxybutyrate. Using structures, discuss reaction sequence of synthesis of any 5 glucogenic amino acids. If you talk about the synthesis, at the time you are looking at the degradation, you will see the arrow was going towards synthesis and degradation, especially at the point you're talking about the aspartate. From oxaloacetate, it will form aspartate. And then aspartate, you just add an amine group, it's going to form asparagine. For glutamate, you could say, from alpha kg, it forms glutamate, then glutamate to glutamine. The glutamate dehydrogenase was involved somewhere there. For alanine, the same. So it's a bit of reverse, but then the enzyme is different. Bypass steps in gluconeogenesis, we've discussed that using structures. Substrate level phosphorylation using structures. Discuss substrate level phosphorylation in glycolysis. This question, if I ask the student, during the time I was asking the student confused this step with ATP utilization steps. But it should be ATP generation steps, because the substrate level phosphorylation we are talking much about here involves the step where phosphoglycerate kinase is involved, and the pyruvate kinase. Those two steps. Step number 7 and step number 10 is the substrate level phosphorylation steps. Using structures, discuss reaction sequence for beta oxidation of fatty acid. Specifically, it should be even chain fatty acids. So I need structures for those 4 steps where you have the FADH2, the NADH, the hydration, the thiolysis, but it has to be the way it is, sequential. So it has to be step 1 where the NADH is generated, step 2 where hydration takes place, step 3 where generation of NADH, step 4 where thiolysis or cutting of our fatty acid, activated fatty acid to carbonless, where we generate acetyl-CoA, and a fatty acid which is 2 carbonless. Discuss, am I repeating myself? Discuss reaction sequence of ketone body formation. We discussed that. Using structures, discuss ketone bodies formation is the same one. Discuss reduction steps in fatty acid synthesis. I told you, by the time you're synthesizing fatty acid, the oxidation which we had in beta oxidation now becomes reduction. So only the reduction steps is what you need to discuss here. There are also 2 steps where reduction takes place in the fatty acid synthesis. Only those steps. And then another question says, using structures, discuss phosphoryl shift step in glycolysis. Phosphoryl shift. I said phosphoryl shift is the same as mutation, where we have the phosphoglycerate mutase. A shift from the 3rd to the 2nd, where the 3-phosphoglycerate now forms 2-phosphoglycerate. That step, give me that step, you'll get your marks. Then I say again that using structures, discuss phosphorylation, activation, and epimerization step in galactose metabolism. What I'm asking here is simple. I want you to show me, using structures, the steps where the galactokinase is involved in the phosphorylation step. The uridyl transferase is involved in activation by addition of uracil or uridine. The last steps where epimerization is involved. Because remember, we said galactose is an epimer of glucose at carbon atom number 4. And the enzymes are named according to the reaction they catalyze. So the epimerase will catalyze the conversion of galactose 1-phosphate to glucose 1-phosphate. So show me using structures and the enzymes. You'll get your 10 marks. How does ketosis result? This one I repeated several times. Using structures, outline the non-oxidative phase of the pentose phosphate pathway, where the interconversion of sugars take place, where we have the transaldolases, transketolysis, the epimerases, the isomerases. That's okay. Using structures, show the anaerobic and aerobic fates of pyruvate. We said the aerobic fate will give us the acetyl-CoA. And then this acetyl-CoA can go to the TC cycle. In anaerobic, no oxygen, it will form lactate, which is also used for a source of energy for the cardiac as well as the brain muscles. But then, still, in anaerobic state, it forms ethanol, the fermentation which takes place in those yeasts. So under anaerobic and aerobic, just use structures and put the correct enzymes. We'll be able to see on what the product is. Using structures, show the activation steps or step for fatty acids and draw the carnitine structure, showing its role in beta oxidation of fatty acids. So you just show the step where we are adding the CoSH group, because it's like 2 steps. The enzyme involved there is a step where the kinase will provide energy, and then there's a step for activation. Then you draw the structure of carnitine, this protein which transports the activated fatty acid from the cytosol to the inner mitochondrial membrane for beta oxidation to take place. I think that's it. I'm at number 40. So I thank you for your time. Though some people have also already left, but then time has gone. So thank you so much, and you can revise well for the cut, and wish you well. So Betty, our cut, we said it's on Tuesday at 10. It will only be 10 marks for 20 minutes. So that is agreed. Let us avail ourselves there. Okwanyo, thank you so much. Thank you so much. So let's prepare well for exams. The video, Ochieng, the video is going to appear on my YouTube channel. After 1 hour, you'll be able to see it. That is the good thing with YouTube. It saves it. You know, the other one which you had on Google Meet, you couldn't find it, because even me, I couldn't find it, and I'm sorry about it. But then this one is going to be available. And even with the other ones, which you already revised, so they can help us to prepare well for exams. So I wish you well, and you have a good night, sorry. So thank you so much, and God bless.